Hi everyone, welcome. We'll just wait a little bit of time until you've all joined, and then we'll kick off this this webinar for today. So just hang in there. Numbers are growing. Good time to make sure your cup of tea is warm. Numbers still growing. We'll just give you another 30 seconds. I'll mute my phone. Great. Okay. Well, we'll make a start now. Hi, I'm Theo Brandt. I work at the Immunisation Advisory Centre as what was the Communications Manager, now the Information Manager. And today this webinar session, we're sort of looking at the safety data of the Comirnaty vaccine, that is the Pfizer BioNTech vaccine, the one that we're using in New Zealand. So we're looking at the data both from the trials, from the original trials, and then we're going to sort of carry on looking at the ongoing safety data through the surveillance data that will be collected, from now on, and what's been coming in internationally. This webinar is part of our sort of ongoing vaccination webinar series. And if you do need to leave early, or if you know someone who hasn't been able to attend, these will be available as recordings to watch at a later date. Just note that we do disable chat on Zoom, so you're encouraged to pop questions in the Q&A as you go along if you wish. But there will be more dedicated time for Q&As after our two speakers for today have presented. So just a quick introduction of who we've got with us today. Sort of in the background, supporting behind the scenes with your questions and answers, Sue and Bronwyn from the IMAC COVID team. And then from a presentation perspective, we've got Dr. Emma Best and Dr. Helen Petousis-Harris. So Emma, I'll introduce Emma first, and then I'll introduce Helen when she speaks. So Emma's a specialist pediatrician. She's up in Auckland, a graduate of Auckland University, and a specialist in pediatric and infectious diseases, with more training in Australia and Singapore. She's also one of our medical advisors, which is why she's with us today. Her major interests include the impact of vaccination on child health, and she's done quite a lot of work around rotavirus, pertussis, measles, and varicella and pneumococcal diseases. So Emma, we will hand over to you at this stage.
Kia ora tātou, and thanks Theo for that introduction. I hope that my screen sharing is working.
It is.
Thank you. So I'm going to do the kind of whiz-bang overview on COVID vaccines, I should say, which may be familiar, I'm sure will be familiar to many of the people who are listening, and then Helen's going to give us a bit more in depth on how we look forward to monitoring these vaccines in the future. So I'm a paediatrician, but I do work with IMAC as a medical advisor, and we're really getting up-to-date information continuously around the COVID vaccine program and rollout. So just very briefly, you know that it's an ongoing pandemic in the world. We've still got daily hundreds of thousands of cases happening across the world. This is up till mid or early March, and still having deaths in the tens of thousands. But I think New Zealand, as we know, is in a really privileged position because we have had cases in the tens and hundreds. Our epidemic has really gone away, and we've actually had controlled borders, so we really have very few cases happening, and that gives us time to reflect on what we know about the vaccines as we roll out the vaccine and how important it will be to help open up our borders and open up the world. But we have that time and that information, which I think is really critical, and we're really incredibly fortunate, as we know. So I think the way out of this pandemic is with vaccines as part of our global control. So so that's really what this is all about for us. So the interesting thing for New Zealand is we actually have got, instead of it being a COVID vaccines talk, we've got a one COVID vaccine at this stage as our primary control strategy. And just briefly about the virology of the COVID virus. It's a single-stranded RNA virus, so it's got its RNA in the middle with some capsule around the outside and spike proteins on the, that are important, and they're in particular important as a vaccine target, but they are also the thing that helps the virus enter the cell and binds to those particular ACE2 receptors that are pretty widespread in the lungs and other respiratory tissues. So when we think about the actual vaccines we have available, we can just talk about the one, and that is the mRNA vaccine that we're using in New Zealand. And that is specifically the mRNA CV vaccine, I call it, but it has got quite a few different names. And I think we hear about the Pfizer vaccine, which is the main company that's producing the specific vaccine. It's got a brand name of Comirnaty, which is a funny combination of the word community and RNA to create this brand name. It was called this long acronym for the clinical trials, and I tend to think of it as its method of action. So it's a messenger RNA type vaccine against coronavirus disease. But you'll hear all of these used potentially interchangeably if we talk about the coronavirus vaccine or the COVID vaccine that's being used in New Zealand. So it is an RNA vaccine, and what that means is the RNA fragment that encodes specifically the spike protein of the coronavirus is encased in a lipid coat, and that's what's injected into the body. That mRNA is taken up and used, the human cells will create the proteins, the virus proteins, the spike proteins, that are then expressed by the immune system, or what we call the antigen presenting cells, to the immune system to give us the immune response. So the components are that fat bubble that has the mRNA inside it that gives the instructions, or a part of the mRNA, the instructions to our immune system or our cells to create those proteins to express to the immune system. So I'm going to put some sort of frequently asked questions sort of throughout my talk, because we commonly get asked about, are these mRNA vaccines things that will change our own DNA or in a person or within a person's cell? And no, that's not possible. The RNA vaccines don't actually integrate with our genetic material. They don't even enter the nucleus where our DNA is contained. They're taken up by the cell, and they give instructions to our cellular mechanisms to create the antigen, that spike protein, that stimulates the immune system. After that, the RNA, the message is actually broken down pretty quickly in the cell cytoplasm. So the mRNA vaccine, CV vaccine, is given by 2 doses, 21 days apart, and given intramuscularly. So it just has that very small amount of nucleoside mRNA within the lipid bubble, and the coat has polyethylene glycol, or PEG, on the outside, as well as some cholesterol and some phosphocholine to create that lipid coat. And these are important components of the vaccine. Otherwise, there's just some salts and sucrose, just when you're thinking about if there's any allergy issues, to know what the actual lipid coat is made from, as well as the contents. So it's designed to make the body produce the virus proteins. It's not actually active or replicating in itself. So it can it give you COVID? No, there's no risk of the vaccine causing vaccine. It's not causing disease. It's not a live vaccine. It doesn't replicate. I've certainly had this question quite recently. I've had the vaccine, just yesterday. Does the vaccine cause a false positive if I go to have a COVID swab? No, that the sampling we do from the nose is detecting live replicating virus. It doesn't give you a false positive or anything like that should you have the vaccine the day before. How is it made, and what kinds of human cell lines are used? Well, actually, there aren't any human cell lines used in the production of the vaccine. It's made from a DNA template in big, large vats, and they use specifically designed bacteria to multiply replicate to create the mRNA message, which is then broken down by different chemicals. The actual manufacture and vaccine don't require any contact or use any human cells or live virus specifically. And they use chemicals and enzymes to break these into the building blocks to create the vaccine. These are images from the laboratories in the Pfizer laboratories in America that were in the media earlier in its production times, and these are the freezers where it's stored. This new technology seems to be really fast and happening. We've got this happen within a year where these steps were skipped. Well, no steps were skipped. The genetic sequence of the SARS virus was made very quickly available by China, actually, in January of last year. And the technology that is used to make this vaccine has actually been used already to investigate other vaccines. There was a huge amount of funding, obviously, because it was a pandemic, mobilised quickly so that they were able to do things sort of simultaneously in vaccine production and development, including upscaling factories at the same time that the clinical trials are being rolled out. So there haven't been steps or safety steps skipped in this vaccine manufacture. And I think that's important to help reassure people that this hasn't been fast-tracked or done in any kind of dangerous way or differently from other vaccines that we use. So looking, as I was saying, we're in a lucky position because we can look back at what the world's already done. And you can see, I've just updated this from 400 million, but there are 550 million vaccines have been given. Not all of the Pfizer mRNA type vaccine, but that is a large proportion of them across many countries in the world already to this date. And different countries are using different formulations. These are the countries, there's 80 countries using the Pfizer, the mRNA CV vaccine that New Zealand is using. Also, it's being used in Australia along with another type in Australia. Many countries in the US, Canada, and Europe are also using this vaccine. So just a little bit on what we know so far about how effective and efficacious it is. And efficacy is something you might observe in a clinical trial where they have a randomizer or placebo-controlled group. And then there's the next step, which is effectiveness when it's put out into a population of people who are at high risk of disease or of different ages, sizes, and those types of things. So we've got both efficacy data and real world effectiveness data now. Again, New Zealand's in a privileged position to be able to have all this information already. So the original important randomized control trial was started middle of last year, and the initial efficacy results were from 43,000 people, quite a very broad range of ages, and it included people with comorbid medical conditions and those who had high BMI. Up to 1 in 4 or 5 people had obesity or BMI of greater than 30. They were in a randomized controlled trial, and they weren't looking specifically at how effective it was against asymptomatic infection. It was only just about confirmed symptomatic cases, and it had a very high efficacy. And similarly, it had a high efficacy of around 88 to 90% against severe coronavirus disease as well. So that's the efficacy data from the trial. And since then, we've had multiple studies reporting what's happened as they've rolled it out within their countries. And so here's a couple of those. Scotland, where there's been 1.1 million people who've received one of the mRNA CV vaccines, and the other half of the population received the other formulation, it should be AstraZeneca. But after a single dose of the coronavirus Pfizer vaccine, 85% of people were effectively prevented from having hospitalisation at a month after the vaccination. So that's just after the first dose. And importantly, that effect was equally good. That high effectiveness of protecting them from hospitalisation from coronavirus disease was as good for those elderly people, those over 80. More information from a large number of people, 62,000, who were what were called high-risk population, including those healthcare workers and rest home residents were at high risk of severe or symptomatic disease and were being exposed after 2 doses, really good effectiveness to prevent symptomatic coronavirus disease. And this is just from Israel, who've used only the Pfizer vaccine, showing that it has extremely good effectiveness against documented infection, that is PCR positivity, symptomatic coronavirus disease, being hospitalised from coronavirus disease, having severe disease as reported by different types of levels of need of healthcare and death. So really good effectiveness data with, really people in the millions being reported now from this vaccine. When we think about the other vaccines that we actually use and you compare that effectiveness in the real world that we're already seeing with these mRNA vaccines, it compares really well. So we know that the influenza vaccine across different age groups, potentially in the elderly or those who have immune compromise, it's less effective, but it's still a really important part of our armament against influenza each year. So far, the coronavirus vaccine is looking to be more effective across a wider age range than the commonly used flu vaccine. Hepatitis B, chickenpox and measles vaccine all have very good efficacy and effectiveness, and yes, the numbers are similar to what we're seeing with the mRNA coronavirus vaccine. And other vaccines that aren't are really important for disease control, but may wane with time or be not so effective as you with time, like whooping cough, are also commonly used in our schedule. So the kind of information we have about this vaccine would really stack up well against things that we use now for controlling important diseases. So what's briefly the New Zealand coronavirus Cova vaccine strategy? We know it's about getting those who are most at risk of severe disease protected first, and those who are most most risk of being exposed to the disease being vaccinated first. It won't be mandatory, it's not available for private purchase, and anyone who's in New Zealand, regardless of their immigration status, will be eligible to have this in the appropriate sequencing. So we know that we've already moved through border and managed isolation quarantine facility workers and the family that they live with, that frontline workers and people with high risk, living in high risk settings, and that includes those in the greater South Auckland area and Counties Manukau, or those with who have eligible conditions and are older, will be considered in this group 2 group. And then moving along in April, May will be people who are at higher risk of serious disease, and finally the general population. As a paediatrician, I often get asked about whether the children will get the coronavirus vaccine or COVID vaccine. Well, I'd say no, not yet, but I don't think it's off the cards by any measure. It's going to be an important part of strategy 2, potentially. The vaccines, the current COVID vaccine is only licensed from the age of 16 years and older, but there is data ready to emerge, I think, about the children who receive this vaccine from the ages of 12 to 16 that have already been happening overseas, and there's trials for those younger as well. This is the New Zealand up-to-date information from as of March 24th. So you can see we've delivered 41,500 vaccines already in New Zealand, and hopefully a good escalation to cover the whole population, target population, by the end of the year. If you've had, it's much, at least a common question in New Zealand, but if you've had coronavirus or COVID disease before, do you still need a vaccine? Well, yes, we don't know if the immunity after COVID infection is perfect, or certainly how long it lasts, and it looks like the vaccine will be an important part of protection for people even if they've had coronavirus disease. The studies have actually recruited those who've had COVID infection, those who haven't, and the vaccine efficacy was equally good amongst those who had or hadn't had disease. And you don't need a blood test to say if you've had it before. There's no issue if you have possibly had COVID before, you can still safely receive the vaccine. But who can't have this COVID vaccine? Well, there's very few absolute contraindications to vaccination. Essentially, it's only if you've had an anaphylaxis to a previous dose of the coronavirus vaccine, or potentially a possible anaphylaxis, proven anaphylaxis to a component of that, which would include the PEG component. So those people should be referred to an allergy specialist for discussion before. There is a precaution that if you have had anaphylaxis type reactions to other products or vaccines, that you should be given a longer period of observation following that, and pregnancy is a precaution at this stage for the vaccine as well. But those who are on any different medications or blood thinning types of medications can sort of proceed with vaccination. And those who are pregnant and at high risk of COVID exposure should also consider this. So just more specifically, obviously common questions around the safety of the mRNA CV vaccine in pregnancy. It's, again, not a live vaccine, so there aren't expected risks to a mother or fetus. It's not going to replicate within the body or transmit across the placenta. At the moment, it's a precaution, and that's because women who are pregnant weren't actually enrolled in the efficacy studies. But it's important if you are at risk of exposure, that is being a border worker, being a healthcare worker, that you do go through the process of considering having this vaccine in pregnancy. Otherwise, you can defer until after delivery or after birth. And no need to avoid getting pregnant if you've had the vaccine, no need to pre-test people to see if they are pregnant before giving the vaccine, and it's got no issues around vaccination and fertility, and no issues for administering to women who are breastfeeding. And there is actually real world data emerging all the time about women in pregnancy who have been vaccinated, whether they have a role as a healthcare worker or they're living in countries that are having higher rates of disease and they've elected to have the vaccine. So to the end of February, there were over 16,000 women in the US who had received the vaccine whilst pregnant. They're being actively followed up, and there's been no safety concerns reported to that date. And I tried to look at that this morning again. There wasn't any further information, but some real world data showing no adverse outcomes for pregnant women who've received the Pfizer BioNTech vaccine in the States. One of the other considerations that's come out around this vaccine is, does it have a higher rate of anaphylaxis? That's sudden serious allergic response happening after the vaccine. It does look to be slightly higher than what we see with other vaccines. Probably when we talk about childhood vaccines, there's a chance of maybe 1 in 2 million to up to 1 in a million chance of an anaphylactic type reaction. So it's still very uncommon. And it looks like from information evolving, and this is from the US information, that it may be up to 5 per million doses may have a serious anaphylactic response. That's still very, very uncommon, as you can see. Importantly, amongst looking at those cases more closely, a third of the people who ended up having anaphylaxis reported that had that to something else before. So there's possibly a flag in their story, which is why having had a prior anaphylaxis is a precaution for this vaccine. And almost all of the people had these symptoms occurring within the 30 minutes, which is why that 30-minute wait time is really important if there's been prior anaphylaxis to any other agents. Other frequently asked questions, being, as I talked about, being on blood thinning medications like warfarin. Yes, you can have the coronavirus vaccine in those situations. It can be given to people on different types of anticoagulants, including aspirin, heparin, and ticagrelor. I can't even say those as a paediatrician. Other FAQs, can I have the vaccine if I'm on immunosuppressants like azathioprine or methotrexate? And yes, you can have that, even those of people who've had prior solid organ transplants, those who are on specific types of checkpoint inhibitors, including things like Keytruda. Yes, all those people are able to have the vaccine without issue. It may be that the immune response, if you're very immune suppressed, is not as good against the vaccine, but it's not a safety concern as such. And some people will discuss with their oncologists if they're on active treatment, the best time to get the vaccine for them. Will we still be having an influenza vaccine? Well, for healthcare workers, yes, the influenza vaccine program, and for those at risk will start in mid to late April. It's likely that for some people the COVID vaccine rollout will be prioritised. And so, for example, the DHB where I work at the moment, we're trying to get that done before the influenza vaccine program. But we'll see how that rolls out. Can you have other vaccines with the COVID vaccine? Well, there isn't any data at the moment about simultaneous administration of vaccine. And so to minimise any confusion about associated or adverse events after the vaccine, the advice is 2 weeks on either side of giving an mRNA COVID vaccine. So giving that, trying not to give any vaccines 2 weeks before or after. And for a live vaccine, for example, MMR, waiting for up to 4 weeks before giving the COVID vaccine would be important. But there isn't a problem if you need to actually give another vaccine. For example, someone who's tetanus exposed and they've recently had a coronavirus vaccine, then do just give them their tetanus requirements if they need that, regardless of the gap. How will I feel after my vaccine? Well, actually, my friend who's a GP in Brisbane sent me this when she got her vaccine because literally I hear they've had a few cases there. So yes, for many people, it's actually going to be something they're really wanting and looking forward to as part of our way out of the coronavirus COVID pandemic. But actually, yes, adverse events are quite common. It is so the reactogenicity, it's a vaccine that might cause a bit of pain at the injection site. It's pretty well tolerated. These side effects all seem to be within 48 to 72 hours. There are systemic symptoms that seem to be more commonly reported by younger people, the younger age group, than those who are over 55. And most resolve, as I said, within 2 to 3 days. And there was quite a high use of antipyretics or analgesics after having a vaccination. And here's some of the real-world data looking after first and second dose of the COVID vaccine, the Pfizer COVID vaccine in the States. Here's 700,000 people who've been actively asked how they felt after their vaccine. And you can see that injection site pain is, yes, very common after the first dose. And there's fatigue, headache, aches and pains and chills happening on day 1 and day 2. But actually after the second dose, even more so, more of the fatigue and headache and the aches, more common after second dose on the first and second day. So again, this is 200,000 people, but up to 40 to 50% reporting that they felt pretty fit and tired, had a headache after the second dose. So it is reactogenic, but the symptoms certainly resolving, almost gone by day 5, 6 for, well, almost all people. And here's some real world data again, I've just updated this morning from Australia. Of those people in Australia who received the mRNA CV vaccine, they actively asked them via text messaging, how did you feel after that first dose? And 2/3 said didn't feel any problem at all. And after the second dose, 40% said they didn't feel anything at all after that second dose. So more closely looking at those who did feel not so good after the first dose, mostly again, injection site pain, feeling tired, headache, body aches. less than 1% of those people actually needed to see a doctor or go to an emergency department in the days after vaccination. After that second dose, the numbers are smaller in how many people have actually been actively followed up because they're still rolling out the campaign. Yes, more people did report they didn't feel great, and up to 2% did actually have to go and see a doctor about how they felt. But it was, again, those things we're expecting: fatigue, pain, muscle aches, headache, joint aches, and low-grade fever. So yes, it's reactogenic, but not severe. How long after getting the vaccine is until it's effective? So the mRNA COVID vaccines do need 2 doses. There is immunity after the first dose, and the best protection is most likely to be up to 2 weeks after the receipt of the second dose. And here's again some real-time effectiveness information just released today, looking at 4,000 healthcare workers in the United States. It was effective in preventing COVID, so they were being repeatedly exposed to COVID. They were 80% effective 2 weeks after that first dose of the vaccine, and 90% effective 2 weeks after the second dose. So really good real-world effectiveness data for high-risk healthcare worker groups being tested every week after their coronavirus vaccine to see if they've got disease. What about the vaccine's activity against the COVID variant strains? Well, yes, we know there has been emergent, whilst the pandemic is wildly circulating, the virus has been mutating and changed, and most of those mutations happen in the spike protein, which is that important vaccine target. These more transmissible strains are potentially also more virulent, although I'd have to say that it's kind of conflicting evidence around that. And the vaccine efficacy or effectiveness against the variants is sort of emerging and perhaps a little unclear. There's some laboratory evidence that it might be less useful, and in other animal models that show that there is protection from infection using the vaccines. But there's now some real world data. So we have information from the UK where they looked again at their frontline healthcare workers who are getting the Pfizer vaccine. They looked at how they tested them very frequently after their vaccination, and overall had a reduction in infection after the first and second doses. And what's interesting about this information is this was whilst they had the UK variant strain circulating. So that was the predominant strain circulating. And yes, we're seeing actually quite good effectiveness still after that second dose through this information from the UK, where they've got the UK variant as a predominant infection. So it did also look at asymptomatic infection, and that means they were testing people to see if they had positive tests without any symptoms. And that sort of information really helps us think about what's the future with regards to how this might impact on the herd immunity. Here's some other information, not the Pfizer vaccine, but the AstraZeneca vaccine, which was used in a randomized controlled trial in South Africa. 2,000 participants randomized, and using, unfortunately, because of the South African variant strain, with this AstraZeneca vaccine, the efficacy was only 10%. So unfortunately didn't show good protection against mild to moderate COVID in South Africa against the South African variant. And that's not the same vaccines we're using in New Zealand, but it does use that same spike protein antigen. So as I say, the data is kind of emerging and probably a little bit unclear at the moment. And this is just a summary from WHO, sort of collated information about the 3 other vaccines there is some information about. These are not the ones we have in New Zealand, but there is a future that we may have another vaccine as well. What's their efficacy against variants? Well, against mild to moderate disease, Novavax didn't look too bad at all, 85%. As I showed you before, AstraZeneca against the South African variant, only 10% effectiveness. So there are other vaccines that are being used in the world that are looking to have some efficacy against the variants. And I think that may be the way of the future for us. So lots of questions still. How long lasting will the protection of the vaccine be? Will we need to have boosters? And I think that's part of the conversation around those variant strains. If I'm vaccinated, will it stop me from spreading the virus to others? Well, I think that's important herd immunity information that, again, I think is coming out. So just very briefly, the last information that I have is about this asymptomatic infection, which is what we need to help us understand about the chances of herd immunity. So in Israel, they've done an amazing job of vaccinating a large number of people, but they've targeted their healthcare workers, and again looked very closely at how those vaccinated healthcare workers, what was their infection rate. So not if they had symptoms, but if they were asymptomatically infected, and it was 75% effective in stopping any kind of infection after just one dose. So I think both the Israel information and the UK information, and now what's just come out today from the US, showing that those healthcare workers who are being watched for any kind of infection afterwards, is quite good information showing that the Pfizer vaccine is causing reduction in all types of infection. These reducing asymptomatic infections will actually be part of what gives us herd immunity. So I think I've just gone through my first half hour. I hope that wasn't too quick, but I will leave it at that and let Helen talk more about how we're going to look to safety in the future as well.
Great, thanks very much, Emma. We will carry straight on just in case we get a bit of time for questions at the end. So now I'm very pleased to introduce Dr. Helen Petousis-Harris, vaccinologist, associate professor in the Department of General Practice and Primary Healthcare here at the University of Auckland, and the director of the Vaccine Datalink and Research Group. Since 1998, she's been involved in immunisation-related research in New Zealand, including clinical, social science, epidemiological, and health systems. Her main research areas are vaccine safety, vaccine effectiveness, and currently co-leading the Global Vaccine Data Network. And Helen, take it away.
Well, thank you, Theo. Hey, is this all showing okay?
Yeah, perfect.
Oh, good. We press the right buttons. Okay, so congratulations. We have COVID vaccines, and of course our journey doesn't end there by any means. And here we go. We've just got to make the thing advance. There we go. So what are we in for in terms of safety challenges? And here's me being creative, but on the right, you can see a couple of headlines. These ones are from the one at the top is from the US and the New York Times, and it's reporting on the appearance or reports of thrombocytopenia following administration of the Pfizer vaccine. And the one below, I think we probably saw appearing here at the time, and this was reporting on the deaths of some elderly and quite frail residents in aged care following sort of soon after receiving the COVID vaccine. Now, with respect to the elderly deaths, there was no assessment of the potential causality, concluded there was no causal relationship there. And I think there's some reasonable satisfaction that at the moment there's not an issue with thrombocytopenia, but it is one of those conditions that is being kept an eye on. So, and on the left is something of my own creation, because I think, you know, we've lived through these things many times, and what we can expect and what we can anticipate is that there's going to be reports appearing in the media of adverse events appearing after immunisation. And we'll just click that again because it's not, here we go. And in case there was any doubt in your mind right now, this is not real. This is fake and misleading headline. Fortunately, this has not happened. But anyway, how do we make sense of this? Because unfortunately, we are going to see these things happen. They're happening now. They're happening a lot overseas, these headlines, and they will happen here. And I think it's good to go back to a little bit of, I suppose, history and experience to help us understand what to expect. And this is a graph that was developed, or a chart that was developed in the late 90s by Bob Chen from the CDC. In his effort to explain the evolution of an immunisation program. And what we see is we see the presence of a disease. We always start with the disease, of course, and it prompts the development of a vaccine. And when the vaccine comes into use, people are usually quite keen to take it up, and the coverage goes up. With increasing coverage, the rates of disease begin to decline. And then you start to become more acutely aware of adverse events, and these might be real or perceived events. And these, of course, serve to perhaps shake the confidence of the vaccine program, and you can get a decline in the coverage here. And with a decline in coverage, you often see a resurgence of disease. We've seen this many, many times, particularly measles played out across the world. Now, realizing that this disease is actually horrible, we're reminded that this is not nice. There's a resumption of the confidence in the vaccine program, and hopefully with its resumption, we will see the control or elimination of the disease. And to remind ourselves that this is where we are here now. We are very early on in the evolution of an immunisation program. There's a lot of things that are likely to play out in the future. And one of our challenges is being able to separate the legitimate events from those that are coincidental. So the example I've got here is, if there were 10 million individuals vaccinated in the UK, just by chance, you'd expect to see, say, 21 or 22 cases of Guillain-Barré syndrome and 5 or 6 cases of sudden death within 6 weeks of vaccination, just by coincidence. And if you wanted to do an assessment and see if the vaccine increased the risk of these things, you would need a study that had about 4 million people in, just to detect a twofold difference. And this is something that's really hard to communicate, because it can be quite hard for people to accept the idea of something being coincidental. That can pose a lot of challenges. And then but beyond those events that are very rare, we also have to consider that vaccines and people are by no means all the same. We've got, well, in New Zealand at the moment, we've just got the one vaccine to worry about, but by and large, we could see others in the future. And globally, COVID vaccines vary. There's all sorts of formulations out there in play now. The populations that we're using the vaccines on vary considerably, and that's not just for example, geographically and different racial and ethnic groups, but also we're using them across different age groups, as well as people with a whole variety of underlying conditions. And then the way people access healthcare varies, and also the way healthcare is delivered varies. And of course, well, let's have some seasons as well into that very complicated mix. So it does pose some challenges for assessing safety of any vaccine, but right now assessing the safety of COVID vaccines and the safety profile of a vaccine is really it's a body of knowledge. It's the result of a lot of information coming from a number of different places. And first of all, of course, we start with the clinical trials that Emma's just taken you through. And these are, I mean, they're really considered the penultimate, because usually they're randomized and have significant, I mean, they've got some big numbers in them. They need to be big enough to show that the vaccine prevents the disease, of course, and also provide enough evidence of safety to satisfy the regulatory agencies in the first instance. Now, there's no way you're ever going to be able to do a trial big enough to measure rare events. It's going to be too expensive, and it's going to take too long. And the other thing you can't do is measure, and you can't measure indirect effects. You can't estimate community immunity, and it would be very difficult to assess the effect on transmission during these types of trials. Often you don't have genetic diversity in these trials, although there has been quite an effort to include that in the COVID trials. But one might, from our perspective, there's actually very few Polynesian trial participants at this point, although New Zealand's going to be doing some sort of phase 2 type studies to include Māori and Pacific people, to ensure that there's, some nice data collected there as well. Because we also know from our own work in New Zealand that ethnicity is actually quite strongly associated with vaccine responses, and that's not necessarily in a bad way. Actually, our observations have been that we can actually have some more positive, some better responses to vaccines. But You know, you can't assume all people are the same. So, yeah, clinical trials have their limitations. So what are the solutions to that? And we've got a lot of questions that we need to answer after we start deploying vaccines. So the first, and I guess the most widely spread tool that we have is passive surveillance, which is something that's been around a long time, the spontaneous safety surveillance. In New Zealand, that's through the Centre for Adverse Reaction Monitoring. If it's Australia, it's the TGA. And in the United States, it's the Vaccine Adverse Event Reporting System. And I would flag the VAERS system as being something that crops up a lot in anti-vaccine arguments, and it's always something to be aware of. It is a passive reporting system. So these guys all recruit or all collect voluntary reports of adverse events, and actually most countries have one of, you know, varying quality. And also most countries contribute their data to the WHO, the Uppsala Pharmacovigilance Monitoring Centre as well. These are very good for signal detection. When issues in the past have arisen around vaccines where there have been genuine safety signals, these systems have been very good at picking those up, because they do pick up things that are unexpected and unusual, but they can't tell you anything about causality. You cannot use these for causality assessment. So let's get a little bit more active about the way we assess vaccines and look at enhanced surveillance. And because it's 2021, we use digital technologies to actively collect events that can happen post-vaccine administration. Now, in Australia, they've been using these for quite some time. They have SmartVax and VaxTracker over there. And New Zealand has adopted the SmartVax and made some modifications to fit with our needs here. Now, as far as I understand, that has not yet been activated. So it was ready to go, but not activated. But hopefully that will be, if it hasn't already, it will be very soon. And what these do is can reveal events that aren't going to be picked up by a GP visit, because people don't necessarily visit their GPs for everything, of course. It's nice because you can involve the patients in the pharmacovigilance process. But again, like the passive surveillance, you can't use these sorts of things to infer causality. And also, it's not going to be helpful for complex or rare events. You don't have any unvaccinated people here, so there's no comparison. You can't do any comparisons between vaccinated and unvaccinated people. Now, last year, the meeting at the Global Advisory Committee on Vaccine Safety, the WHO, one of the, it was all about COVID. And one of the things to come out of that meeting was to highlight that the most urgent immediate task for most countries is to establish the normal background rates for adverse events of special interest. So there's lists that have been developed and of events that we might want to be looking for very hard, very actively to sort of ensure the safety of these vaccines. Not every country can do that, but countries like New Zealand can. And where we really ultimately want to get to is being able to compare very large numbers of vaccinated people with very large numbers of unvaccinated people. So this is where we start getting into the really big data. Now, that can be at the national level, or it can be site-specific. If you're in New Zealand, we've got exceptional data, so we can do the vaccine safety monitoring and subsequent studies are second to none. So that's all very well, but our population is itty-bitty. We've got a very tiny population. So when you start thinking about the numbers that you need to look for very rare events, you're not going to have enough people. So this is where you need to go even bigger. So the team of 5 million is great, but we're going to need some more people. And this is where you can get into distributed networks. So if you can imagine those dots, even though they're in different configurations, each dot is either a country or a site within a country. And if they're all using the same methods, the common protocols, then that makes it possible to bring them together in like a type of meta-analyses. So you can really almost have these mega global studies. And while different countries and different sites might have slightly different systems, if we all use the same approaches using the same definitions, then we can combine the findings together. The European Union have got one of these now operational since COVID. They've been working towards it for some time and managed to get that operational. The United States have had one for quite some time. But really, there's been a couple of exceptions where truly global studies like this have been conducted, but it was never really sustainable. So these are great, and we've always traditionally looked to the United States for a lot of our really robust, large safety studies. But I don't think we should assume that they've got all of this covered all of the time, because COVID is something that we are all in together for. And it requires everybody to make contributions. And in fact, just last month, it was highlighted actually in the media where people were calling for more effort as the US systems were struggling to get up and going to full capacity, to the best of their capacity for this COVID rollout. So you can't always assume that everyone else has got this and you're just going to be able to rely on what they're doing, especially when you have the capacity to do it very well yourself as well. So it just brings me to the ultimate solution, which is going back to those dots, going back to globally distributed networks. And what we have today, as of this was launched in January 2019, the Global Vaccine Data Network, and that has 17 countries with around 21 sites at the moment, with a population of coming on 300 million people. And these sites all have the capacity to do this, to use their large administrative data sets to deploy common protocols in order to be able to share the data, to share the findings, bring them together to look at rare events. And there's a list of the current partners. So that's to highlight some of the tools that we have to be looking at the safety of these COVID vaccines. And I guess the main points are that safety scares have the capacity to derail these programs. It's always happened in the past, and I think we can anticipate it happening now. And I think we can see it happening. In fact, look at the issue in Europe at the moment with the concerns around the AstraZeneca vaccine and blood clots. We've got some really good preclinical data, and that gives us some optimism that these vaccines are going to be pretty safe. And certainly the Pfizer vaccine looks like it's got an exceptional safety profile at the moment. So we've got really good tools to continue to monitor it and continue to look for really very, very rare events. But I think we're in a vulnerable space at the moment where the world has to get this ramped up and ticking along really well. But I think quite soon we're going to be able to have some very, very strong sort of data, very good studies on these vaccines. In particular, the Pfizer vaccine, also because it's being so widely used. And I think I will leave it there.
Great, thanks so much, Helen. Now we do have a couple of questions that have come in, so I'll just hand over to Bronwyn and to Sue to manage those with our two presenters. Thank you, Bronwyn.
Thank you for those great presentations. So the first question I've got here, if the adult patients are lighter or have a lower BMI than the 16-year-old people, do you still recommend the same dosages? And will they experience more adverse effects?
Who do you want to answer that, Emma or?
I think it came up during Emma's presentation.
Yep, no, I'm happy to say that there's no dose adjustments for this vaccine, so it's still the same dose for anybody, any size, 16 to 85. Younger people generally, in the big picture, when we talk younger, 16 to 55-year-olds, reported more reactogenicity, so adverse events like muscle aches and those types of things after the first and second dose than older people. So I guess that hopefully will answer that question, but I can't say that BMI, low BMI or lighter than an average adult was would make any difference to that.
Thanks.
And the other question here is, if you have a positive COVID test or have had disease, how long a wait before you can receive the vaccine? So you can still have the vaccine if you've had COVID in the past. And possibly one of you ladies might like to jump in with active infection of COVID.
Emma wants that one too.
Emma wants that one. Sorry, I'm on now.
Yeah, so I mean, if you're not unwell with a fever or actual active infection, then you can have the vaccine at any time. I think there's a little bit of information that some people are waiting for 6 months after the infection, but that's not been recommended in New Zealand. Certainly you can have it, if you haven't got active infection or active illness, you can have the vaccine anytime, and it doesn't matter if you've had COVID in the past. As I say, we still need to give those people the vaccine to protect them for the future, because we don't know how long-lasting infection from infection is, or how long-lasting protection from infection is. So we do want to vaccinate people who've had COVID before.
Another question we've got here, what were the results of animal trials on the vaccine?
I'm not quite sure what they're asking, but if we're thinking about the animal models that where the vaccine's been tested on, there are a variety of animals used depending on what you're trying to do. Like if you want to induce an infection, you need an animal that will actually get COVID infection, as opposed to if you want to look at the antibody with the immune response, you want an animal that mimics a human immune response, and all the animal studies, and of course it doesn't move into humans until you've got to demonstrate in the animal that you can raise the appropriate type of immune response that you know is going to be desirable, and that immune response can ideally protect against disease when you challenge the animals with the disease. And also we've got data to show that when the animals were challenged, that the vaccine was able to prevent quite well the infection in the upper respiratory tract as well in the animals. So if that's, hopefully that answers that question.
They used macaque monkeys, didn't they, for some of those trials? We certainly use them for respiratory viruses because they are similar to humans. But a lot of human data, I guess, which is what's most useful.
Thank you.
Can I repeat?
Did you want me to do that one, Sue? You got it.
Oh, that's all right. So this one is, can you repeat the statistics for New Zealand's COVID vaccinations after dose 1 and dose 2?
So I'm not,
I guess
sure.
That means
so we don't have symptom, we don't have symptom information post-COVID vaccine 1 and 2 that we're actively getting. That's right, isn't it, Helen? We don't have actual active, we're not monitoring people actively. That will be optimum, but I have got that data from Australia, which is on their VacSafe website. I don't know what it's called, Vax Tracker. And the actually stuff has the current number of doses that it's recording. So you can actually Google stuff, vaccine tracker for New Zealand, and the current number of doses we've given is 41,500 that I could see from March the 24th. I don't know how many of them are second dose. We've started doing second doses in the last week, so I'm sure that perhaps some of the vaccinators here will know that information, but I don't have numbers on dose 2, and we haven't got active surveillance on their symptoms. But the Australian data, you can go back and look, but it was showing that 65% of people weren't reporting any adverse event or feeling of unwellness after the vaccine, the first dose, and about 30 to 40% weren't reporting any problem, but about after the second dose. But that means about 60% were feeling not so great after the second dose. But the symptoms were again that mild and not lasting fatigue, headache, and muscle pains.
Next question we've got here is, how can the public have reassurance that the vaccine has been stored correctly and safely right up to the time of injection? I know we have very strict cold chain processes, and everybody that's been giving the vaccine is trained very well in these processes, and cold chain leads that are overseeing this at every site. If anyone wants to jump in and add any more to that.
Yes, all the sites are cold chain accredited, and the people that are working in those sites have received cold chain training. So yeah, it's a fairly tight process from my understanding.
Yeah, and I'm certainly on the back at the end of the phone, myself and other medical advisors at IMAC and all the really expert immunisation coordinators whenever there is a concern or something that may be a problem, and that's immediately responded to by. So there's, I would say feel it's a very good system at the moment.
Mm.
Looks like one for you next, Helen.
Yeah, I can see that. Are there any concerns about enhanced disease associated with this vaccine, given there have been no long-term studies? This is the idea where you have a vaccine and you develop say an antibody response, and then when you come across the disease, because you don't have the right kind of, shall we say, antibody response, it can enhance the disease and make you sicker. So this has been seen before in the past with some selected vaccines. So this was something that's been particularly looked at, and particularly before it went into humans as well. So we've got a lot of animal data on this, and there is absolutely no indication that that's going to be the case. They've tried to make this happen, and that's because the nature of the immune response is doesn't lead to that. And then the human data and the immune response in the humans is also suggesting that that's not going to be the case. And now we're a little bit down the track too. We're not seeing it. And the other thing is, when you take the sera from people who have had natural infection and you give that sera to people to protect them, you don't see enhanced disease. So that's another one. So I think we're probably going to be okay on this one. And it is an event that is one of the events that's being looked for in particular. But so far, there's no sign that that's going to happen.
Yes, I'd agree, Helen, because antibody enhancement is looking quite good, that there's not an issue. I've been asked about long COVID in the vaccine, and I don't know if you have any theory on that. The overall long COVID, the feeling of being unwell and fatigued after having actual COVID disease, will be less when we vaccinate, because there's less actual active disease happening. Whether the vaccine can cause long COVID, it doesn't seem that it would be biologically plausible, because it's not a live virus, immune stimulus, but I guess, and there was actually just a report on a woman who had long COVID and got the vaccine and felt better afterwards. So yeah, I would say that that's not going to be a concern. I think more importantly, getting rid of the disease to prevent long COVID is going to be much more important.
Yeah. Yeah, I even heard I did hear that some people who had long COVID received the vaccine and then actually appeared to be a lot better. So I'd be interesting to follow that one as well.
Okay, well, everyone's getting very keen on questions all of a sudden, just as we're almost out of time. So I think, well, let's go through the ones that we've got there now, and we might need to call it a day at that point.
There's one here, you may be able to help, Bronwyn. Is it true that you have to return the vials from COVID vaccines so that no one can fill with water and try and pass it off? Now, look, that I have not heard anything like that to date.
They are definitely very on site. They are not letting any of those vials out of their site at all. So they're very carefully monitoring those vaccine vials after they've been used to ensure that they don't actually leave the site.
Yeah, I'd agree with that.
Yeah. Okay, and Helen, did you want to have a look at that one about the flu vax? So we're just saying that last year VAERS data for flu vax had 30 related deaths in USA.
Mm.
How many COVID-related deaths would concern you? I mean,
well, first of all, I would say that those flu deaths are not necessarily caused by the vaccine. They've been reported as events that have occurred following the vaccine, not necessarily caused by the vaccine. I want to make that very clear. And anyone pulling that data from the VAERS database, you can go in there and you can pull it. Before you go into the database, you actually need to acknowledge you understand that these are not necessarily caused by the vaccine. These systems are early warning system, they're signal detection, but they can't tell you about causality. So far, there's not been a death that has been attributable to the Pfizer COVID vaccine. And we, at the moment, there's a lot of attention on the blood clots that may be associated with the AstraZeneca vaccine. So yeah, if we're talking about Pfizer vaccine, to the best of my knowledge, that has not occurred. And please be very wary of passive surveillance data. It can lead us astray and is often sort of spread around as
it's often
misleading.
Yeah,
misquoted. Yeah.
Okay, well, I think we've probably reached a natural conclusion to today. So again, I'd like to sincerely thank Helen and Emma for their presentations today, to Bronwyn and Sue for managing the questions, and for everyone for attending. And just remember that if you do want to catch up on bits you might have missed, we will make this recording available probably within the sort of the next 4 to 5 days, but with Easter being where it is, no guarantees. Thank you very much for your time and have a great rest of your day. Bye-bye.